Let's learn regarding average bioequivalence, population bioequivalence, and individual bioequivalence. These terminologies are used for the bioequivalence. These are abbreviated as ABE, PBE, and IBE. So B is for bioequivalence, and it involves the different studies like average bioequivalence, population bioequivalence, and individual bioequivalence. You might be knowing about the bioequivalence. What is bioequivalence, what is the exact meaning, and why it is done. So bioequivalence is the study to compare the two formulations, how they behave in vivo, and bioequivalence studies compare the rate and extent of the drug absorption, which is denoted by Cmax and AUC. Bioequivalence compares the two formulations in the individuals, which may be the patients or healthy subjects, for the comparative evaluation. Bioequivalence study involves studying the drug behavior or the drug kinetics or pharmacokinetics in the individuals. So, in this video, we are going to learn regarding three types of bioequivalence studies like average bioequivalence, population bioequivalence, and individual bioequivalence. I was also unaware or I was not having sufficient information and knowledge about these three terms. I am also learning, and I am very happy to see that by making this video along with me, many of the professionals are also learning. Let's start with this video, and I request you to be with me till the end of this video to have this good idea about the bioequivalence studies. See, generic formulations require to demonstrate the bioequivalence with reference formulations. And you can also say there is a regulatory requirement to demonstrate bioequivalence to get the generic approval. Also, whenever there are some changes or variations are made to the existing formulation, then that time also, if the changes are major, then bioequivalence studies are required to be performed. Average bioequivalence study, by definition, we can say that average BE is a standard method used to compare the pharmacokinetics parameters of the test drug versus reference drug in the group of subjects. We know about the studies like crossover study, parallel study. So in this, the group of subject is made, and with this group of subjects, the parameters of pharmacokinetics like Cmax and AUC is studied. So one group receives the test formulation and another group receives the reference formulation, and then the pharmacokinetics are compared. This is the simple meaning of average bioequivalence. This average bioequivalence ensures that on average the two formulations are bioequivalent, or two formulations pass the B criteria. Key aspects of average bioequivalence is based on the 90% confidence interval approach, that is 90% CI approach. The test to reference ratio should fall within the standard limit of bioequivalence, that is 80 to 125% for Cmax and AUC. And this average bioequivalence study does not account for variability between the subjects or within the subjects, because it is based on the average of two formulations for the Cmax and AUC. So this is the generalized study, or this is the very popular study for proving the bioequivalence between different formulations. The formulations may be generic also, and the formulations may be the change or formulations with some variations than the original approved formulation. When B study is used, or when the average bioequivalence study is used, so it is the most widely accepted bioequivalence method for the generic drug approval. It is done for various types of formulations, maybe immediate release or extended release formulations. These may involve crossover studies or parallel studies. So whenever you are coming across the bioequivalence requirement for generics, always consider that it will be a average bioequivalence study. Now let's move to the another important study or type of bioequivalence, that is population bioequivalence or PBE. Definition of PBE is it focuses on the total variability. As we saw, average bioequivalence doesn't involve or focus on the variability, but population bioequivalence focus on the total variability. Total variability including the within subject variability and between subject variability for both the test and reference formulations. And as it is a study for total variability, it ensures that distribution of the drug or drug exposure across a population remains consistent. So population B study, as the name indicates, it studies the distribution of drug exposure across population. Key aspects of population bioequivalence are it compares variability in the entire population between the test and the reference product, and it uses statistical methods involving variances of PK parameters. So it is not commonly used in routine generic drug approval. In a generic drug approval, routinely the average bioequivalence study is used. So when population B is used, so it is used for the drugs with high variability in the population, and it is used to evaluate biosimilars and narrow therapeutic index drugs. There are some drugs which are categorized into NTIs or narrow therapeutic index drugs, like immunosuppressants are there, some antiepileptics are there, some antihypertensives are there, which show the narrow therapeutic index. So for those, population bioequivalence study can be used or is required sometimes. Then come to the individual bioequivalence. It is IBE. Definition of IBE is it ensures that individual receiving the test or reference formulation experiences similar effects with minimal within a subject variability. So it is individual level bioequivalence. As we saw, average bioequivalence is among the group. Population bioequivalence is for the total population to study the variability, and now individual bioequivalence is for ensuring the variability within subject. So it is a stricter than ABE, as it assesses whether the individual can safely switch between formulations. So you can understand here that individual safety is considered in the individual B. Key aspect of individual B are it accounts for subject by formulation interaction and differences in how individuals react to test versus the reference. Always understand here that it is an individual level bioequivalence. It uses within subject variability to ensure consistent drug exposure, and more complex statistical analysis is required compared to the ABE. Now, when ABE study is required for drugs where switching between the formulations would cause adverse effects, like antiepileptics are there, like phenytoin. Just for example, I am giving you the antiepileptic example. Immunosuppressants are there, antiarrhythmics are there, and it is recommended in narrow therapeutic index drugs. So population B and individual B, these are recommended for the variable drugs and NTI drugs. Now see the differences between ABE, PBE and IBE. ABE focus on the mean PK parameter, PBE focus on population level variability, and IBE focus on the individual level variability. Statistical method for ABE is based on 90% CI, which should be within the limit of 80 to 125%. PBE variability comparison is there, and IBE variability plus subject by formulation interaction is studied. ABE studies only within subject variability. PBE studies total variability within plus between subjects, and IBE studies the within subject plus subject by formulation interaction. ABE used for generic drug approval to show or demonstrate the generic drug is behaving similar to the reference formulation when tested in vivo for the pharmacokinetic parameters. PBE is used for biosimilars and highly variable drugs. IBE is used for anti-eye drugs, patient safety focused drugs, because IBE complexity is very high. Now coming to the complexity of study. So ABE study is simple. PBE study is a moderate complex, while individual bioequivalence study is the most complex, or it is having high complexity. Now concluding the video or video information, ABE is most commonly used for bioequivalence of generic products for approval. PBE ensures population wide similarity and is useful for biosimilars and highly variable drugs. IBE is the strict method and is ideal for anti-eye drugs where switching could cause significant clinical differences. This was the information for ABE, PBE and IBE. I hope this information might be new for many of the professionals, and I also think that whenever we are working in the field, we should know the terminologies so that whenever it is asked, we can answer, and as a professional we should have knowledge of these things. Thank you for watching the video, and let's meet soon with such kind of informative video. You can learn from my videos in different languages. You can use the subtitles, and you can learn in your native language, or you can learn in the language you most understand easily. Thank you.